Hello you guys, welcome back to another video on my channel, The Science King, and today we are going to be talking about enzymes. So what are enzymes? Enzymes are catalysts produced by living things. A catalyst is a substance which increases the speed of reaction without being changed or used up in the reaction. Living things have thousands of different chemical reactions going on inside them all the time. These reactions need to be carefully controlled to get the right amounts of substrates. You can usually make a reaction happen more quickly by raising the temperature. In living things, enzymes act as biological catalysts. Enzymes are all proteins and they work all the same way to catalyze various reactions. For example, DNA replication, protein synthesis, and digestion. DNA replication is where enzymes help copy a cell's DNA before it divides by mitosis or meiosis. These are two types of cell divisions and I will be covering them in future videos. Protein synthesis. Enzymes hold amino acids in place and form bonds between them. Digestion. Various enzymes are secreted into the gut to digest different food molecules. So variables that can affect a enzyme controlled reaction, and there are three main variables. The first variable is the change in temperature. Like I mentioned in the first two slides, temperature, if increased, can increase the rate of reaction. So the second variable is pH. All enzymes have an optimum pH at which they work best at. It is often universal pH 7, for example water. The third variable is substrate concentration. This also affects the rate of reaction. The higher the concentration, the higher the rate of reaction. So enzymes have special shapes. Chemical reactions usually involve things either being split apart or joined together. The substrate is the molecule changed in the reaction. Every enzyme has an active site, the part where it joins to the substrate to catalyze in the reaction. Enzymes are really picky and they usually only work with one type of substrate. For example, amylase works with starch and so on. This is because for the enzyme to work, the substrate has to fit into the active site. If the substrate's shape doesn't match the active site shape, the reaction won't be catalyzed. This is called the lock and key theory, and to the side here we can see an example, an image in which we have a substrate and we have an enzyme and how they fit together and are either joined together or split apart. So measuring the rate of reaction in an enzyme controlled reaction. You can measure the rate of reaction by using amylase as the enzyme and starch as the substrate. Amylase catalyzes the breakdown of starch, so you can time how long it takes for the starch to disappear. To do this regularly, take the drop of amylase and starch mixture and put it onto a drop of iodine solution on a spotting tile. Record the color change. It will turn blue-black if the starch is present. Note the time when the iodine solution no longer turns blue-black. The starch has then been broken down by the amylase. You can use the different times to compare the reaction rates under different conditions to make the experiment more reliable. So that's it for today's video, you guys. Hopefully you enjoyed. Make sure to subscribe, like and comment, and I'll see you in the next one. Goodbye.